Hey, howdy, hey folks, it is Mrs. Johnson from the Agro Wisdom channel. We're talking real agricultural genetics today, which is great because we've been leading up to this for like 4 or 5 videos now. And I'm pretty gosh darn excited to talk to you guys about this, because this is my favorite part now about understanding genetics, is actually understanding how the parts and pieces move in agriculture, which is nifty, and also leads to cool things like I forget what they're called, but closely related to a lot of the data that you get from breeding programs, particularly for like meat production, which is really cool. I'll do a video on it, I promise. Don't know how high quality of a video, but it'll be attached to this list of genetics things. Anyway, back on topic. Real agricultural examples. First and foremost, I wanted to pull this out of my bag of tricks to show you just the mass variety of genes that we can talk about when we talk about agriculture, even in like something as simple as a pea plant. That can be fairly simple to understand when you just look at each individual piece, but can get more convoluted as you add genetics or add more conditions onto it and more genes and traits onto your list of things that you want to talk about. Pause, look, do your things, use some examples if you want. Practice, do your thing. Next one, just an example of a Punnett square using the pea plants. If you wanted to practice, you could practice. Just draw squares, assign parent genes, and see what comes out of it. Just do the practice. It'll be fun. It's worth it. Just practice the simple stuff. And then you can also jump into things like practicing the harder stuff and running through all the questions. You know, what genes do the parents have? What gene options do the children have? What would the next generation look like? What do the ratios look like? And just kind of running through as you practice and seeing things. Or even just like looking at your own pea plants and kind of gauging that. Looking at your own assortment of things, whether it be cattle, horse, dogs, sheep, cats, house plants, non-house plants, garden plants, whatever it is. People, even. There's a lot of simple gene things like earlobe attachment, widow's peaks, chin dimpling. I think these dimples are also fairly simple genetics to kind of look around and play with and study and experiment with. But yeah, practice. And once you learn how genetics works and kind of do a nosedive into what this information can do for you, you can nerd out for days about it. That being said, we can talk about things like codominance in roan cattle. Right? We have roan, and you can see we have red and white genes, R and W. R and W make roan cattle. Two red genes, or 2 red alleles, my apologies. 2 red alleles makes a red cow. 2 white alleles makes a white cow. A red and a white allele makes a roan cow. And if you were to cross 2 roan cows, some people that don't know genetics might go, oh yeah, you'll just get a bunch of roan cows because they're the same kind of cow color. You'd get 2 roan cows. Kind of like if you have blonde parents, you're probably going to be blonde. Isn't always the case, as you can see by the genetics. You have a 50% chance of getting a roan calf and then a 25% of a red calf, 25% of a white calf, based on that Punnett square, which is cool and fun and interesting. Another example from the cattle world is polled. Polled is having horns or not having horns versus having horns. In this case, polled cows are dominant, which is important to this thing. You can also, if you're practicing, do stuff like this and do fun little charts like this. And I will actually be working from the bottom up because it makes the most sense in my brain on how I explain it. If you want to pause the video and just see what you can come up with, that's great. Good educational tool. True polled, when you breed two true polled together, the only genes that they can feasibly have is the true polled genes. When you breed two homozygous recessive, in this case, so two horned cattle that only have horned genes, they would only make horned cattle. And this goes into, you could deep dive into, like, breed history with all this crazy stuff. Specifically, horns versus no horns. Has a lot of cool cowboy history. And then, what happens when you breed a true polled and a polled? 50% true polled, 50% polled, heterozygous polled. True polled versus horned, you end up with 100% heterozygous polled. And then when you breed two polled together, like we saw in the last one, you'll start to see patterns. When you breed two heterozygous parents together, you end up 25% homozygous dominant, 25% homozygous recessive, and 50% heterozygous polled in this case, because we're looking at true dominance. And then when you breed a polled and a horned, you get 50% polled, 50% horned. I know this true dominance, quote unquote, is a little touchy-feely in some breeds and some species, because you can be polled, the heterozygous polled, and still show a little bit of horn, but it won't grow very well. So there's stuff like that makes it a little bit flexible in terms of phenotype, the physical representation of the genotype, which is the genes, the letters that we're talking about. And we're moving on. Let's see. This is actually how some color breeds work in terms of dogs. And when you get into horse breed or horse colors and even more advanced dog colors, you see this a lot. We're dealing with two genes. We're dealing with a color gene and an extension of that gene, or a color modifying gene. So, big B, little b is the main color gene. That determines whether it's a black or brown dog. Now, if the locus is the extension, and so if you do not have that E in your gene code, has a organism, you will not be red or yellow. It is recessive, so you can have a big E, and then you're just black. You just stay black if you've got big E's. If you've got little E's, you turn red or yellow. If you've got little E's and you're brown, you're red or yellow with a yellow nose. So you just get lighter with this extension. It doesn't change the dark it doesn't change. How am I supposed to put this? Your locust B, your main color gene, is not affected per se by your extension. You are either black or you're brown, and you stay that way. But you can be a lighter version of that black or a lighter version of that brown based on if you have the extension. If you have the extension. So, when you start looking at colors, you might assume that there's 4 color options. And you might go, oh yeah, there's a letter for each one of those. No. There's just 2 letters, right? A big B, a little b, and then a big E, little e. And you have to be fully recessive in order to be red or yellow. And then how dark of a red or yellow depends on your original gene code. So big B or little b. Which is kind of cool and interesting. Hard to conceptualize without some practice. So I do recommend that you practice. And without further ado, we're going to look at all sorts of dog colors right now. Now, I haven't spent a whole lot of time with this chart. Haven't spent a whole lot of time in recent history on color genetics, so I might be a little fuzzy. But it gives you kind of a idea. So you've got that dominant black and that that non-dominant black, right? So the big letters, you're either black, big B, or you're big B and another letter. And those lead to these options. If you've got little e's, you're yellow or red. If you've got the B extension, if you've got the little b, you're brown. If you've got the D extension, you're gray. Now, this is very brief and, oversimplified, right? And so if you've got 2 little k's instead of a big K and a little k, you have the option to be these colors. The yellow, the gray and yellow, the black and tan, and then different kinds of black and tan based on your extension genes, and then recessive black, and then brown and and then different colors based on those extension genes. And this looks a little convoluted, but we'll jump to the next slide, which makes it a little easier. Now, you'll see that we jump over to horse genetics, which is a little convoluted, but we have 2 locust genes. We have locust A and locust E. I don't know why they're called locusts. We can figure it out. We have Google, though. I'm pretty sure it's because the locust gene is the primary factor into, or at least my best guess, the locust gene is the primary factor into what color you can be. Versus an extension gene is something that affects the locust, but doesn't totally change it like the locust does. So, locust A, if you've got 2 A's, you're black. If you've got an A and an E, or a big A and a big E, you're a bay. If you've got 2 little E's, you're a chestnut. And then basic colors, genes for color modification is listed next. So cream, champagne, dun, roan, silver, dapple, gray. Those are all color modifications. those change how black, bay, and chestnut look. You're still black, bay, or chestnut, but they change the shade of black, bay, or chestnut. Is the easiest way for me to describe it. And then you've got genes for color patterns in horses and in dogs, and in lots and lots of other animals. So Appaloosa, Tobino, Leopard, Blanket, Appaloosa, Paint, Pinto, Piebald, and then your white dominant, I assume, blank dominant, which changes not the shade of color that you are, but where your lack of pigment, quote unquote, where your white comes in, which is cool and interesting. And it's convoluted, I know. Just practice. You'll get it. Practice. This is just an example of how the dun gene on those locust genes, right, that top black, bay, chestnut, and we're looking specifically at the dun gene, this color modification gene, right? So if you do not have a dun modification, you're black, bay, or chestnut. Assuming that you have no other color modifications or pattern modifications, you're just going to be black, bay, or chestnut. Which is simple, pretty, great. If you do have the dun gene and you're black, you will be gorilla. If you're bay and you have the dun gene, you're a bay dun. If you're a chestnut and you have the dun gene, extension gene, you will be a red dun. Which is interesting. And then we also talk about, do you have 2 copies of the dun gene? And it looks like there are, 3 variations of the dun gene, right? So if you've got 2 copies of the dun gene, you've got the dun dilution, which is like a lightening of the color, and the dark, I forget what they're called, the dark extremity points. And you've got some mane and tail colors in there. So if you've got 2 copies of the dun gene, it will be passed off 100% of the time, and you will have the dun dilution and the primitive markings. If you've got a D and an ND1, you will express both. You won't pass it on 100% of the time, because you can pass either. But, okay, cool. And then ND2, same thing, has an ND1. Dun dilutions express primitive markings. As you can see, looks like the big D is dominant. When big D's there, everyone's a dun. What happens with the ND1, ND2 stuff? ND1, homozygous, you won't be done, but you might have primitive markings. If you've got an ND1 and NB2, you're not done, but you might have primitive markings, which is like the zebra stripes, and the dorsal stripe, I think, is a primitive marking. And then if you've got ND2s, no primitive markings, you will not be done. So ND2 seems like the most, like ND2, if you've got ND2, no done. No chance of done at all. ND1, kinda. You got part of it. ND2, or just big D, you've got the whole shebang. You've got done, no matter what color you are. Which is why it's important. Which is why it's, one, if you're breeding for color, important to gene test your animals, because you don't want to breed things a bunch of times and put other lives through the stress of pregnancy without knowing your chances, right? If you want a dun, you want a dun baby, and you've got a dun mare that you want bred, you need to know what kind of dun she is, right? Is she an ND1 dun? Is she an ND2 dun? Is she a total dominant dun? Or what is it? Homozygous dominant dun. What is she? If you're trying to breed her, you need to know that information. Because if she's homozygous dominant, you don't have to necessarily worry about the baby getting the dun gene. Baby's gonna get the dun gene. Baby's gonna be done. But if she's either one of the other two options, she could pass either the dominant gene or the non-dominant gene. If she passes the non-dominant gene, these two, and you breed her with someone that also might not pass the dominant gene, you're going to end up down here. Which means you're not going to get a dun baby. You're going to get one of these guys. And you wanted a dun baby. Which means you should do your research. And it's expensive, I know. But if your heart is set on a dun baby, get your animals bred. Or gene tested. There's places out there that will gene test your animals. And then you can know for sure that you will get that dun baby. That being said, this is just an example of those extension genes again. Or those, not those extension genes, those locus genes. And I like the way this is done because it shows you if you breed a dominant E together with all the options. So a dominant E, heterozygous E, and recessive E, what the options are. What happens if you breed it a heterozygous parent with any of the three options. And it gives you the gene, not only the genotype ratio, but it also gives you the phenotype ratio here. Well, the phenotype, not necessarily ratio, because these two would be considered the same phenotype. Therefore, phenotype ratio 100% for that breeding, right? And then same thing here. Phenotype ratio here, 50-50. Genotype also 50-50. But up here, right, phenotype, genotype have different ratios. Because genotype would explain this big E little e, this heterozygous pairing, has a different option than the phenotype. The phenotype doesn't see any difference, but the genotype does. So, without further ado, I have spelling error on this page. Dang. What did you learn? Drop it in the comments. Learn some things. What did we learn? Color genetics can feel complicated. Taking it one allele at a time. Practicing is important. And it can do some really cool stuff for us in the world of agriculture. And yeah. Have fun. Have a great day. Bye.